So yeah, today I'm going to be talking to you all about my dissertation research, what I've been doing for the last 3 years. Yeah, it's aptly titled "An Analysis of Bird Biodiversity in and Among Isolated Wetlands Through Space and Time." I had a buddy's dad say that more aptly should just be called "Bird Watching Around Puddles." Yeah, here we go. So yeah, I'll give you a brief personal background. Then I'll try to introduce kind of broad brushstroke my dissertation research. Then we'll kind of go through a chapter by chapter. We'll do kind of like a mini wrap up following each one, and then hopefully we'll finish with kind of some big takeaways. Then we'll do some thank yous, acknowledgments at the end, and then hopefully, if I've got this time correctly, we'll have some time for some Q&A. So yeah, as Corey said, I come from a kind of non-traditional background, and so I grew up overseas mostly, and when I moved back to the U.S. I went to a small little school called Barratt College, which is in the Hudson Valley in New York. I was originally a global international studies major, which is kind of poli-sci-ish. I fell in love with birds my senior year and realized that that's what I really wanted to do. But it was tough for me to get a job. And so I took a job as an AmeriCorps rep up in Augusta in Maine. I actually worked on invasive plants. I did forest inventory work for about 6 months. And then after that, I managed to get a job in New York. I worked for Audubon, New York. I mostly did an education program a la carte in all 5 boroughs, mostly teaching kids about birds. We counted a lot of robins. After that, I went back to Barratt and did some more behavioral research, kind of as a post-grad. And then I managed to get myself into grad school. I went to the College of Charleston. And that's where I met Dan McGlynn, who was really fundamental in changing the way that I think about ecology and kind of trying to become a community ecologist, I think, really by and large. And then after that, that led me here, where Corey took a chance on me. Yeah, and so here we are. So to start, so biodiversity. So really, at large, when we're trying to do this, we're really thinking about patterns of species richness, community composition, and species turnover. And for me, this has mostly manifested itself in kind of thinking about how these things change in kind of 2 dynamic ways. And the first one is across space. So how does biodiversity, how do communities change as we kind of move across space at varying scales, right? From just with Florida to the whole United States to across the globe. And secondarily, kind of through time. So that's obviously as it says, it's community composition kind of as the world has changed dramatically over time. And so tracking change in biodiversity, right, like I said, species richness, turnover, community composition, kind of what these communities look like, has traditionally been quite difficult, right? Because we've been stuck trying to count birds, for example, manually across these huge swaths of land and across time. But this has really been changed due to kind of the proliferation of big data. And so eBird and iNaturalist, lots of community science programs have kind of enabled us to think about how biodiversity is changing kind of through space and time, especially pertinent in kind of this ongoing crisis that we find ourselves in. And similarly, we've also kind of had this proliferation of big data products as well as satellite remote sensing. And so all of a sudden now we have kind of real-time data telling us about how land cover is changing kind of by day to day and how bird community composition is changing from day to day. And so with this real-time ability to track biodiversity change, we can really think about local changes. So this is everything from, how does the bird community in Gainesville look as we move from park to park? But it also enables us to really think about how macroecological patterns are unfolding. Right? We can think about how bird communities in Brazil might be changing as deforestation is ongoing. We can think about the entire country kind of at large. And kind of at the center of this biodiversity change, right, that we've kind of all been hearing about, right, all these 3 billion birds that we've lost, and biodiversity is on the decline. Really, at the center of this is anthropogenic change. So humans are modifying landscapes, and it's changing the way that organisms can essentially exist on the landscape. And a lot of this comes down to kind of land cover change. So we're converting forests into agriculture, we're converting forests into pasture, all of these different things. And then we also have natural resource managers that are kind of like managing these resources for different human utility, right? Whether this be timber, for example, kind of managing these different landscapes, managing biodiversity in a way that's kind of beneficial for us. And then this third piece that's going on, right, which is kind of not relatively recent, but in terms of human history, relatively recent, restoration. So trying to actually improve how different habitats look so that they meet these kind of biodiversity, these functional needs that we know that they need. And so these three things kind of interact, at varying degrees across the landscape. And so kind of at the nexus of biodiversity and anthropogenic change, I like to think, and my whole dissertation has really taken this approach, is really thinking about wetlands. And so I've been fairly obsessed for the last 4 years with these things, and I've been particularly interested in these things called isolated wetlands. And these are mostly because they're global in scope. They occur all across the entire world, and they tend to look different depending on where they are. Right, so we have everything from stormwater ponds that are essentially they're built for water and flood storage. They're also used by birds for a variety of needs. Then we have our natural prairie wetlands in Florida, for example, and these are natural. They essentially were formed during the last glaciation when all the glaciers receded. They created these small depressions where water can accumulate. And then everything from things like cenotes are also isolated wetlands. So these are formed when the limestone essentially fell through, and what used to be Florida was a shallow ocean. So now we have these small isolated ponds that are essentially built up underneath limestone. And so they can look quite different, and they're all kind of variably impacted, you can probably see, by different human activities. And so they're occurring worldwide, and isolated wetlands have been incredibly important for a variety of rare and endangered flora and fauna. But they're under serious threat, essentially. And most of this is due to kind of changes in the Clean Water Act that no longer protect these isolated wetlands. Essentially, these systems are no longer, their water surfaces are not directly connected to rivers or lakes or other quote unquote waters of the United States. So they're no longer protected at the federal level. And so states have had to kind of take it upon themselves to protect these wetland systems that we know are incredibly important. They've been lost. We've lost about 70% of them in the continental United States alone. Have had to take it upon themselves to essentially manage these resources for both biodiversity but also human utility. They do all of the water and they clean all of our drinking water essentially that filters down into the aquifer. They also provide flood storage, and they filter out heavy pollutants and all these things. And so kind of these three things that I've talked about, really, there's kind of these three big things that have essentially guided me in terms of thinking about my dissertation. And that's really anthropogenic change, like we talked about. So land cover and wetlands particularly, they're kind of changing. Biodiversity, so where are different things happening, what numbers, and wetlands. And so how do these kind of three things really interact to change kind of through space and time? And so at the center of that is me typically looking at a bird. So here's this is a conceptual figure that's kind of an overview for the 4 chapters of my research. And so if we start at the top there, the first thing that I wanted to do was do a systematic review and synthesis of all of the existing literature on bird biodiversity and isolated wetlands. And this was really to try and figure out, what kinds of things have people already done, and where can I kind of lend a hand into the information gaps that exist in this literature. And then secondarily, kind of moving from left to right in the bottom panel there, thinking about how land cover and wetland areas, are impacting bird biodiversity in these wetlands. And so really this kind of question of how scale is impacting what types of birds can occur in which wetlands, and does it matter if a city has been created around a wetland, or if this wetland occurs kind of in a pristine natural environment. And then thirdly, kind of a minor obsession of mine has been beta diversity. And so this third chapter was really thinking about how these bird communities are changing kind of across space that I talked about, right? And does it matter? Are these community compositions more similar if they occur in kind of the similar ecoregion context versus kind of across the entire United States? How do these communities look? And then finally, the kind of the timepiece that I wanted to add was really thinking about temporal beta diversity across a single day. So I became fairly interested in not just these long time periods about how biodiversity is changing, but how are bird communities kind of altering their behavior in these small little isolated wetlands over the course of just a single day. Okay. All right, so we'll start out. That was pretty good, about 10 minutes. So the first chapter, we're going to talk about research synergies and shortfalls in bird biodiversity in the isolated wetland literature. And this one's currently in review of biodiversity and conservation. It's been in review for about 10 months. I'm hoping for a reply at some point. So the kind of the two main questions here with this first paper was firstly, what does the current literature say, just kind of about generalizable bird biodiversity patterns in isolated wetlands, So what have people done? What are the kind of signals that people have found? And then secondarily, are these patterns consistent across the continental distribution of the United States? So have people found similar signals in Iowa as they have in Michigan, as they have in California, as they have in Florida? So what did I actually do? So I used two different search strings on Web of Science and Scopus that I won't put here because they're quite long, I must say. And it was a nested search term set, and we used the Prisma protocol to essentially try and identify as many papers as possible that have done something that has to do with bird biodiversity and isolated wetlands, essentially. So they had to kind of have those three things that I talked about, right? Mentioning of geographically isolated wetlands, birds, and biodiversity. And so we ended up having a ton, about over 2,000 papers that I ended up screening the title and the abstract and the methods for to make sure that it met those three criteria. I ended up excluding a ton of papers from that, and I was left with about 120 papers that I actually read in full, and then an additional about half of those ended up being filtered out because they didn't meet the geography requirements of being inside the United States, or they didn't really do something that was inherently about bird biodiversity, specifically because it only had a single species, for example. And so at the end of all of that, we were left with 61 papers that I managed to analyze. And so as I went through that, we didn't have any kind of a priori expectation. So we didn't go in saying, oh, I'm only going to look for papers that looked at wetland area and how it impacts bird biodiversity. We kind of did a post priori thing where we kind of read through all the papers. We tried to kind of categorize them into themes, if you will. And so these are all the themes that ended up having at least 5 papers that kind of had at least this theme mentioned somewhere in the paper. And yeah, there's some examples in there. And I'll kind of talk broadly about some of the big takeaways from some of these themes. So kind of the first big figure here is really just trying to think about generally some overview statistics about these 61 papers that we found. So there's been this kind of general increase through time, which is very typical because papers are increasing in publication as time goes on. The journals that they came from, there was kind of a wide variety of journals that they came from, but close to a third or a quarter of them came from the journal Wetlands. And then we also kind of looked at the taxonomic groups broadly that people were interested in studying in these 61 papers. We found that mostly people were interested in water birds by and large, but about a third of the papers also were interested in kind of the entire bird community. We also have some individual species in there. There was some modeling that was done for and picked like kind of a few core species, essentially, and so that's why you're seeing those there. And we found that by and large, about 2/3 of these papers really only use 2 different measurements of biodiversity, and that is mostly just abundance and species richness. And so, yeah, you can see here, general increase over time, kind of a lack of metric diversity really in a lot of the literature so far. Most authors just focusing, yeah, abundance and richness. Yeah. So this is this figure was really to try and unpack a little bit kind of the spatial bias in the papers that have been published already in this kind of broad literature. And so you can imagine like a 1-to-1 line kind of going right through the center of this figure would be like, oh, equal representation for a state based on its kind of its total geographically isolated wetland area versus the number of publications that have been published in that state. And you can find that essentially this top right corner ends up being fairly outsized in its representation. That's like the prairie popple region. There was some federal legislation that was passed that requires folks to count water birds up there. And so those end up getting tons and tons of representation in the literature. And you can find that most states, kind of regardless of the number of wetlands that they have kind of this similar number of papers that have been published in them, essentially. And so finally, this is kind of like the mapping spatially of where those themes that I had mentioned kind of occurred in that literature. And once again, we kind of find that like most of the themes end up overlapping in the Midwestern United States. There is some spatial, stuff going on outside of that, but all of those ones that have like one dot, all of them came like a single paper that was looking at how climate is going to impact biodiversity. And so that's why there's like those single dots in this top left-hand corner talking about climate here. So kind of some main takeaways from this paper and what we kind of generally found in terms of like also what those takeaways were from those themes that we had read about. generally found that there was kind of strong support for the species-area relationship. So almost like there was like 19 studies looked at wetland area, 18 of them found a positive relationship. So MacArthur and Wilson, you guys killed it. And then we found that only kind of 2/3 of those studies, like I said, were thinking about abundance and density. And finally, we also found that there was kind of some mixed land cover impacts on a lot of the bird biodiversity literature. So there were some that were showing that urbanness was increasing diversity, some that were showing that urbanness was decreasing diversity. So there's a bit of a clash there in kind of terms of that theme. And so we really kind of identified and kind of used this paper to think about what should we actually do for the rest of my dissertation, these couple chapters that I'm talking to you about. And that's really that we needed to focus on increased spatial scale. A lot of these studies were field-based. They had kind of a low spatial resolution. Taxonomic breadth, like I said, most of these studies were thinking about waterfowl and water birds by and large. And then really almost none of these studies were thinking about temporal change in these communities, kind of through time in any way, basically. So, all right, the second chapter, which ended up being the first chapter that I actually ended up doing. Corey and I chatted about this when we first started. I think before I had officially started as a student, I started with this paper, of course. And this was looking at kind of how land cover and area impact bird biodiversity and GIWs. The main questions for this paper were essentially, like I said, how did land cover and wetland area impact biodiversity in these GIWs across the entire continental United States? And secondarily, kind of are these signals kind of similar across functional groupings? So thinking beyond just taxonomic diversity, kind of functionally does it impact bird groups similarly? So what did we actually do? So we kind of screened through eBird. We went literally typed the word wetland into eBird. We downloaded every single hotspot that had the word wetland in it. And then we systematically went through, myself and Brittany Mason, who was a godsend doing that with me, to make sure that actually these wetlands met the criteria for being geographically isolated. We used a combination of digital elevation models, land cover, water surface stuff. It was a whole smattering, a smorgasbord, if you will. And at the end of that, we found about 207 of these eBird hotspots that met the criteria across the entire United States. So what did we actually do? So we ended up gathering all of our land cover data for these wetlands from Google Earth Engine, and then we calculated species richness from eBird data, essentially, for these wetlands. So yeah, we used Dynamic World, which is an amazing data set. It's got a global 10-meter resolution for land cover. It's actually changed daily, so you can really capture a lot of variation of land cover quite well. And then we threw kind of the land cover measurements in these wetlands at the local scale, so like just the wetland itself, what it looked like, as well as the landscape scale, which is blown up to about a 25-kilometer buffer around these wetlands. We calculated the land cover. And then we threw all that good jazz into a giant table. And then that giant table was analyzed with a couple of generalized linear models. So for that first objective, so thinking about species richness, once again, MacArthur and Wilson, they were really onto something. Species richness increases with wetland area. So you can see those lines essentially are going basically straight up, essentially. As you increase the size of the wetland, you end up getting more species. I'll also point out that like some of these wetlands, you can see some of these dots around the top of the plot. Some of these wetlands are supporting upwards of 300 bird species over the course of a single year. So we're talking about close to half of any of the number of species you can see across the entire United States at a single wetland that are also quite small. The largest one of these wetlands was about 2.5 kilometers squared. And then each of these smaller lines here at the bottom, they look a little bit cramped because of the log scale and the big number of all species up there. But all of those functional groups, essentially, of birds are also increasing as you increase area. So all functional groups are responding similarly as you increase wetland area. This was most pronounced for terrestrial and aquatic primary lifestyles, which is a combination of things, but basically, you can think of it as a way that a bird essentially meets its life history. How does it forage and where does it forage? They're terrestrial, they do it on land. They're aquatic, they're mostly doing it in water. So those were the strongest positive. They also had the most species. So also on this kind of land cover issue here, what's going on in these wetlands? This is an effect size plot. And beyond it looking a bit scary, we just want to focus essentially on these highlighted bars. So if these error bars kind of overlap with 0, it means that there really wasn't a significant effect we found for that land cover class. These ones that are highlighted in yellow were the ones that we found that significantly impacted kind of total species richness. And we found generally that kind of built is bad, basically. So So land cover that's been human modified is bad for bird biodiversity, both at the local scale, so things like creating a walking path around a wetland, as well as at the landscape scale. So the more buildings, the more homes that you have kind of in this 25-kilometer buffer, those also end up filtering out which birds are going to end up being in these wetlands. We also found some kind of other interesting results, namely that kind of local grass cover and flooded vegetation cover in these wetlands at the local scale can actually increase richness. So we can improve kind of what our wetlands look like to increase richness. This tree's landscape signal was a bit odd, but I think generally what's happening is kind of the reforestation that's occurred across the eastern United States is generally having some sort of filtering effect on the bird species that are utilizing these wetlands. Many of which are forested, so you're removing a lot of those aquatic birds that can use those wetlands. Yes, okay, and on to this second objective, which is thinking about those functional groups I talked about. And so I apologize, this is a bit of graphical violence here. But once again, focus on the highlighted words on the left. This is an effect size plot for these 5 different functional groups. And we can find generally that there are some kind of similarities across these 5 that I'll help you kind of sort out here. And that's that all of these functional groups respond negatively to built land cover, right? So I talked about that in that previous chapter. Oh, like there's this mixed result of urbanization, maybe helping diversity, maybe not. We found by and large that it's pretty bad for all functional groups of birds kind of across this entire spatial scale, spatial landscape. We found, interestingly, that these terrestrial and aquatic primary lifestyles that I talked about, they're really driving those overall signals that I talked about. So those blue highlighted terms there on the left, so that grassland cover I talked about and that flooded vegetation are kind of helping to improve the biodiversity of mostly those functional groups kind of in these wetlands at the local scale. Okay, so yes, the main takeaways for these. Once again, strong support for the species-area relationship, right? So bigger wetland is better for biodiversity, and that built land cover is generally poor and impacts richness negatively, both at this local and this landscape scale for all functional groups. And finally, that these aquatic and terrestrial functional groups of birds are really driving this richness signal, and they can actually improve their biodiversity signal in these wetlands by having more grass and flooded vegetation cover in these wetlands. Okay, we're cooking. This is the 3rd chapter. I'm going to apologize in advance. It's the most petty. The methods here are a bit zany, and so I apologize in advance. But really here we're thinking about spatial beta diversity driven by both kind of ecological and sampling processes. But really this is like, how are these bird communities changing at varying spatial scales across the United States? So our main question here was really just one. It was incredibly hard to answer, so we only had one. And that was, is there kind of higher spatial beta diversity at the ecoregion or continental scales? So are birds kind of more different from each other if they're kind of within this same landscape, so the eastern temperate forests, for example? Or are they more different from each other as we kind of increase that scale to, are these bird communities in Massachusetts the same as Montana, the same as California? And before I kind of do this, I'll give you a broad brushstroke on what spatial beta diversity is, mostly because it confused me for about 3.5 years. And so by and large, beta diversity at its simplest is really just a ratio of local to regional biodiversity. And those two things can be defined at lots of different scales, which is quite annoying. But think about it like this. It's generally, if we think about a couple of local wetland communities, each one of these boxes being a small wetland. So this wetland has a couple of birds in it. This one's got some birds as well. This one's got some birds. We have this kind of regional diversity we can think about. So all of the species that we found kind of in each one of these little local wetlands that we have, and kind of what's the ratio of the local diversity compared to the regional diversity that we have in total. So how do each one of these wetlands kind of complement this regional species source? In order to do this, we often we measure beta diversity, typically using an ever-growing number of metrics, which started with Whitaker in the 60s, but now includes Dracards, Simpsons, Sorensens, and I think basically there's a new name every year, none of which I'm going to use actually. So in true PhD fashion, I reused the data set from my last chapter so that I didn't have to go outside and do anything. And this is a spatial map of those 207 wetlands that I had told you about. Each of those colors there is the ecoregion that it falls into, the EPA level 1 ecoregion. Okay, so how did I actually go about like quantifying this local, regional, all this jibber jabber I've been talking to you about? And so this was a conceptual figure, and we can ignore the bottom half for now before we get there and you start snoring. And so we aggregated the eBird data essentially by selecting only a certain season. So we only stuck with the summer, so we could deal with these kind of like temporal impacts of migration of birds. And we summed the abundance data from 5 random checklists at each one of these eBird hotspots in that summer period. And we threw them all into a giant site by species matrix for all of those 207 isolated wetlands that I had talked about. And then we measured beta diversity using this measurement of biodiversity approach that was kind of pioneered by my master's thesis advisor and others. And it essentially incorporates kind of 2 different ways that we could think about quantifying spatial beta diversity. The first is this multi-metric approach, which is this B curve here. And so really we thought about this kind of 2 different spatial extents like I had talked about, right? So the ecoregion and the continent. So our bird community is changing kind of within an ecoregion, and then our bird community is changing across the entire continent. We did this using rarefaction, which is a fairly complicated thing, but essentially you can think about it as there's a giant hat essentially of species that we can pull individuals out of. And then along that curve and the number of individuals that we pull, everything from 2 individuals to 10,000 individuals, how different do those communities of birds look? Start pulling those different numbers, these varying numbers of species. And then we also use this kind of multi-scale approach, which kind of slightly alters that, right? Instead of imagining individuals that I'm pulling out of a hat, I'm pulling entire communities of the wetlands out and comparing them, essentially. Okay. So what did we actually find? So we, in total, we had over like close to 30,000 eBird checklists that we ended up summarizing. And we actually had to go down to 195 of those 207 that I talked about to meet a variety of criteria that I won't bore you with. In total, we had close to 500 species represented across the entire United States. And total abundance was around 4 million birds. So really dealing with large amounts of data here. We found that there was strong hyperabundance across the entire continental United States. So the most highly represented bird was the red-winged blackbird. Close to 90% of all the birds counted were red-winged blackbirds. Lots of starlings. We also had lots of mallards, my wife's favorite bird. Plenty of the Canada goose, which now decides that it doesn't want to migrate anymore. And of course, the purple martin. And so even though that these species represented less than 1% of the total species richness, they accounted for close to 31% of all of the birds that we counted across the entire United States. So let's break down this kind of spatial beta thing that I talked about. And first we'll kind of tackle this ecoregion extent. So how are bird communities changing within an EPA level 1 ecoregion? And so here are the 5 ecoregions that met those criteria of having enough samples that we could actually do this with. And in an effort to not get too into the weeds here, really, once again, the focus here is really thinking about these highlighted points here. And what these points do is essentially they negate the impacts of what we call sampling bias, things like the species abundance distribution and those number of individuals that I talked about. And they try to quantify whether these communities at each individual wetland are actually different from each other. And we found essentially that almost every single ecoregion was about the same. Most of these error bars overlap with one or are very close to overlapping with one, which would signal to us that most of these metrics essentially are telling us that there's very little change in the bird communities as we move from wetland to wetland. And this is probably driven by this hyperabundance of species that I talked about, right? So when you imagine you're pulling these individuals out of a hat, you have to pull so many before you start getting stuff that's different, essentially, from those 5 species that I talked to you about. And this signal was fairly consistent in each of these ecoregions, but also vary quite dramatically in their landscape context, the number of wetlands that they have. You might imagine that deserts could be quite different from each other with such a large distance between wetlands compared to like eastern temperate forests where, out here in Florida, you can throw a rock outside and you can hit about 5 of these things. But then we went ahead and we did this multi-scale approach because we didn't really know what was going on. It seemed weird that none of these metrics seemed to be different. Like, why are these communities not different? Like, how is that not possible? And so we built these spatial-based rarefaction curves. And so once again, we're really looking here at this difference between the black line from this purple line. Purple line being those random individuals pulls that I talked about, which is this, oh, we pull a cardinal, then we pull a wren, then we pull a wren, then we pull an egret, versus this black line here, where we're really pulling the entire community again, right? One community, then this community, and we're comparing them to each other. And we can find here, actually, that there ends up being some spatial aggregation change as we kind of within each one of these ecoregions. So they do look different from each other. The problem is that the hyperabundance of species is really like negating that signal when we try to calculate a metric, especially with 4 million birds. You can imagine like that's so many individual pulls you have to do to try and get something that looks different. Yes, that's the highlighted spark notes. So then we wanted to blow this up again, and we said, okay, so we've done this within the ecoregion. Let's look at how these communities change if we do it across the entire continental United States. And we ended up finding really very similar results to what we found in the ecoregion level, which is this plot on the left, right? Once again, if we think about just that beta C there, it really isn't a ton of change, regardless of the wetland, regardless of where it is spatially. But when we do this kind of with this multi-scale approach, these rarefaction curves actually end up diverging quite a bit more than we even found at our ecoregion scale. So that these wetlands actually do end up being more similar to each other the closer that they are together. So what are the kind of the main takeaways from everything I just said? So there's kind of conflicting evidence for what spatial beta diversity is across the United States, especially considering which approach that you use. And so our multi-metric approach, for example, was heavily influenced by these hyperabundant species with continental distributions. So it was really hard for us to find a signal using just those metrics that I talked about. Whereas our multi-scale approach, which really takes this more holistic view of what a wetland community of birds looks like, really was accumulating samples one at a time. Those end up exhibiting much stronger spatial aggregation. So they look more similar to each other the closer that they are. And really the kind of the main takeaway from this was when I talked about this ever-growing number of metrics that people are obsessed with making in data diversity, we really can't rely on any single one of those metrics to really say what's going on. And oftentimes with this big data revolution that we have, we're really going to have to rethink a lot of the beta diversity analyses that we're doing, because we're dealing now with millions and millions of individuals, not field-based studies where we're dealing with tens of individuals. Okay, we're doing really well on time. Okay, so finally, the time portion. This is time. So I was really interested, like I said, in thinking about daily temporal beta diversity, and mostly in suburban wetlands here in Gainesville. I did this entire study on my bike, much to my chagrin. I did over 400 miles of cycling. To my knowledge, this was the first CO2 neutral field study that's ever been conducted. And so, yes, I counted birds for an entire year in these wetlands, and let me tell you, July was hellacious. So, daily temporal turnover. So, what was our main questions here? So, the first one was, really, are the bird communities experiencing kind of like this change in the community over the course of a single day? So, do we see the same stuff at 7 a.m. versus at 7 p.m. in a lot of these wetlands? And then secondarily, are birds kind of shifting their behavior in these wetlands over the course of the day? And is maybe that kind of the reason why we're finding different birds over the course of the day in these wetlands? So, I went out there. I'm the black silhouette there, and I went out kind of every other hour from an hour after sunrise to 6 of these wetlands, and counted essentially every single bird that I saw for 15 minutes. And we noted the behaviors that they were doing, behavioral scans every 5 minutes, essentially, noting kind of the first individual bird for each species that we saw and what they were doing in each of these wetlands. And so this is kind of an intensive sampling approach, if you will, that's quite different from like an extensive sampling approach, where typically we try to maximize the number of sites that we can get to maximize our statistical power. I was really quite interested in nesting this kind of within day. I needed to know, like, how is this wetland bird community changing over the course of a single day? So these are what they look like. We broke them up into kind of 3 general vegetative and hydrologic categories, essentially. And so they were emergent, open, and forested. These open ones are the ones that we're all familiar with in Florida, right? These giant, like Disneyland wetlands, where it's just a giant pit, fills up with water. There's nothing in it. vegetation-wise, and they're fairly permanent throughout the year. They typically tend to retain water almost the entire year. Some of them are also billed as lakes, so to speak, to the HOA. You got to pay extra to live on one of these bad boys. And then we also have these kind of emergent wetlands, which are kind of A on the left-hand side, which are really like they're an open wetland that basically like the lawnmowers haven't gotten to in a little while, right? So there's more vegetation growing in there. They're probably not managed as heavily. And then our third and final one are these forested wetlands, and these ones have trees, and basically they're forested, and so they're kind of like each of these is like kind of a big step up in terms of the amount of vegetation that's occurring in each one of these wetlands, which might change productivity, might change the bird communities we're finding. So, what do we find? So we'll stick kind of with species richness to start here, and so what this box plot is showing you here essentially is broken up into the breeding and the non-breeding seasons. How is diversity changing over the course of the day? So just number of species in each of these wetlands. And we find generally in the breeding season that the diversity is fairly consistent throughout the day, right? So we're almost always finding at least 3 species in these wetlands, regardless of the time of day. There did seem to be a bit of a steeper drop-off in the non-breeding season over the course of the day. So birds maybe aren't utilizing these wetlands as strongly over the course of the day. But those shaded gray areas are the error bars, and you can see that we really don't have a lot of power to say that it's decreasing, or that it is really truly decreasing throughout the day. Okay. And let's move on to abundance, right? So I did like those first 2 that I said that everybody does. So we did those 2 first. Abundance also kind of fairly the same throughout the day, right? So we're like straight abundance. It's not really changing as we kind of go count these wetlands as the day gets longer. There does seem to be some noticeable dip again in the non-breeding season, but once again, those error bars are quite large. We're not really certain. And there might be higher abundance in these open wetlands. So these ones that are that have no vegetation in them, probably because wading birds tend to congregate them in super large numbers, as opposed to our forested wetlands, which have like very few passerines, for example. But once again, not really super sure about that being a definitive signal there. Okay, now on to the thing that we actually cared about. This is the beta diversity. So the temporal change throughout the day. I struggled a ton with how to visualize this, and this is what I settled on. And this is essentially, if you look at this y-axis here, a beta diversity value of 1 means that the communities are completely different to each other, and a beta diversity value of 0 is they're completely the same. And so the way you read this plot essentially is if you go to each of these months, we compared the community in the first hour after sunrise, which is the most typical time that people go to count birds. And then we compared that community subsequently to each of those other hours that we ended up counting the bird community. And so you can see here that there's tons of variation in how these communities are changing, depending on which of those pairwise comparisons you look at, right? Which one of these colored dots. You see generally that the diversity doesn't, spatial beta, like beta diversity doesn't increase throughout the day, or decrease throughout the day. So it's fairly, it's relatively the same. So the bird community 7 hours after sunrise is just as different from the first hour as 11 hours after sunrise, right? So each of those dots kind of is hovering somewhere around 0.7, 0.8. So about 70 or 80% turnover in that community is what we're seeing over the course of a single day, right? So we're having tons of replacement of new species. And then onto the behavior piece, right? So we were interested, like, so now we know, like, diversity is kind of the same throughout the day. We have the same number of species, but we know those species are changing as we go throughout the day. Is their behavior changing? Maybe that's what's leading it to happen. We found generally that foraging dominates what's going on in each of these wetlands. So they're hyperproductive in terms of all birds are spending the majority of their energy and their energy budget essentially in these wetlands, foraging, looking for food throughout the day. We did find interestingly that there is like this slight change in that month of July that I talked to you about that was hellacious for me. And it was also fairly hellacious for the birds because they were like, I'm not going to go out foraging when it's 107 degrees outside. I'm sitting and I'm just going to like preen my feathers. I'm going to just hang out in these wetlands essentially. So yeah, we can see it. A lot of these lines are fairly straight. There is a little bit of change, but generally foraging dominates. Then we kind of summarized all of those behavioral values over the course of a single day to figure out just kind of like what's the overall total energy input that these birds are doing essentially in each of these wetlands over the course of the entire year. And once again, it kind of bears out the same signal I talked about, right? Like 60 to 80% of the time, birds are foraging in these wetlands. There is this big dip that happens in July, probably because it was so hot. We also did find that there is some breeding behaviors. There are birds that are breeding in these wetlands. By and large, it's very few because the wetlands themselves are so small, they can only support a certain number of breeding individuals, essentially. But we had breeding black-bellied whistling ducks, breeding Carolina wrens, breeding common yellowthroats, kind of a wide variety of birds that were breeding in these wetlands. So, kind of what are the main takeaways here from this this final chapter here? So, the biggest one is really that bird sampling. Lots of birders tend to go out birding in the morning. That's when birds are most active. It's historically when all of the counting of birds happens. It's like between sunrise and 3 hours after sunrise. But for these kind of suburban wetland systems, they may really be ignoring or underestimating the biodiversity value and utility of these wetlands, because there's lots of different species that are kind of coming in over the course of the day to utilize them for some life history need, mostly foraging. Managers, conservation practitioners, people who run their HOA, all of these people should really start to consider not just this like single subset of kind of the birds they're utilizing in these communities, and really think more holistically about the entire bird community over the course of the entire day. Currently, wetland mitigation standards and laws don't require any temporal replication of counting, especially for birds in wetlands where you have to mitigate one so you can destroy one and build a new one. All of that counting only has to happen at a certain time. And our study would tell you, like, well, you probably need to go back multiple times to really fully guarantee you've captured the whole community that's utilizing this wetland. Species richness is consistent throughout the day, right? I told you there's 3 species throughout the day. And that's most important because we found that turnover is super high. So we know that we are missing species as we go throughout the day, right? If beta diversity was super low, we would know like, it's just the same birds that we're seeing over the course of the day. But beta is super high. So we're actually, on average, of those 3 species, 2 of them are different out of every single count, essentially. So this is the, I blazed through that. Good. I talked too quick. Bit of a Yankee, they say. So here's what if you haven't listened to the last 45 minutes of me blabbering, here's what I hope you take away from this whole thing. And that's largely that systematic reviews can help reveal a lot of the synergies and the shortfalls that exist in literature. And in our case, it really showed that there was a lack of spatial evenness in sampling, and people were really relying on only a single metrics to think about bird biodiversity. Land cover is filtering out species at the landscape level and at the local scale of this built land cover. It's really bad for birds. But we can improve the way these wetlands look to increase the biodiversity value with that flooded veg and grassland cover. The communities in these wetlands are hyper-dominated by hyper-abundant species, and they kind of require multiple methods for us to really uncover the signal that's going on in a lot of these wetlands. And finally, fine-scale temporal changes probably deserve a bit more attention in this big temporal beta space, right? We're always constantly focused on how has things changed over the last 150 years, but our wetland bird communities are actually changing a ton just over the course of a single day. And so sampling and sampling regimes really need to try and change to harness that entire bird community that's utilizing these wetland systems. Okay, and now this might be the longest slide that I go on. And so a PhD requires a lot of time, has a lot of effort, a lot of money. And so I have a lot of things that I need to acknowledge. So the first one is my wife who's here with me today, and my daughter, of course, who's made doing a PhD, I think, seem easy, maybe, in some ways. My wife has always called me her long-term investment plan. I've made zero money for a long time, and she's supported me throughout the whole thing. And I'm really fully indebted to her, essentially, for this entire experience. I wouldn't be here today without her. All of my friends and family, there's so many of them. I think a lot of them are on Zoom today. They've all been incredibly important sources of inspiration, laughter, consternation, the whole gambit. And I really am indebted to all of them also encouraging me to kind of go count birds, essentially, for the last 10 years. So I wouldn't be here without them as well. My committee members and my advisor, They've all probably listened to me talk about this stuff, probably way too much, I would say. Maybe too many meetings I've scheduled, I would say. And Corey really took a chance on me when nobody else did. Nobody else even gave me an interview for a PhD. And Corey kind of took a swing on me, and I'm really fully indebted to him for anything that ends up happening to my career. It really wouldn't have happened if he didn't say yes. Both the labs, the GERG lab, so that's Corey's lab, as well as Katie's lab, that's been my home away from home as I've been here in Gainesville. They've let me kind of give a smattering of presentations about a variety of things, both coherent and incoherent, over the last 3 years, and have helped me really improve me as an ecologist, as a researcher, and help me think about kind of birds, biodiversity, wetlands, and everything kind of in a really a totally different way than I did when I came years ago. The White Department, there's so many people here that help cut my paycheck. They help me schedule getting my dissertation defense. They have met with me to talk about beta diversity or a data set that I think I wanted for free. And so I really am fully indebted to a lot of them as well. CALS, they give me all the money that has funded me essentially for the last 3 years. So I really appreciate them. Countless faculty, I've met with like close to 20 or 30 people. Sometimes it was only once, sometimes it was once a week. And all of them have kind of helped me in a variety of ways figure out what the heck I need to do myself. GAU, the union here, who reps for all of our benefits and our healthcare, that helped us have our baby at a fairly inexpensive rate. So I'm fully indebted to them as well. All the eBird contributors, my dissertation wouldn't be possible without community science. So all the people that go out and submit eBird checklists, they're essentially, I'm fully indebted to them as well.
Except the mean checkers, I don't like them.
Yeah, the mean checkers, yeah.
Screw them.
A variety of previous supervisors and employers, I think some of whom are on the Zoom today, who have all helped me do countless things. And mostly paid me so that I can afford to do this stuff, which has always been my dreams, to do ecology and get paid to do it. And all of them have played a part in allowing me to do that. And then lastly, I'd be remiss if I didn't thank the birds, of course, because they're out there and they're letting me count them, and I'm very appreciative of it. And with that, thank you all for listening. I really appreciate it. And this is the published chapter of my dissertation in diversity and distributions. It's open access, no paywall. Go check it out. Yeah, and thanks so much.